Okay, now we're going to talk about how we actually make sure that we did it, how it actually works in the lab, how we put it into the bacteria. So this was like a very simplified method. We already had the plasmid from the bacteria and we inserted the gene, but this is just DNA. These aren't actually in any cells yet, and we can't make an organism do this unless we put this entire plasmid into the organism. So here is an example that I found online that I want to use and like kind of record over. So this one is an example of frog DNA. But in our lab, we're using jellyfish DNA. So just pretend that this is jellyfish DNA and that this purple here is the gene that codes for that fluorescence. So what happens is that we want to take out this gene, right, and put it into something else. So we use restriction enzymes, like we just said. EcoR1 is an example of a restriction enzyme. So we go through and it cuts that gene out. And then we do the same thing to the plasmid. So we have bacterial plasmid and it cuts at this specific site. Okay, so now we have some of this gene and we have some of the plasmid. One thing to recognize is what I showed you here is I just cut the gene exactly. But remember that these recognition sites are just long stretches of like repeated parts of DNA. And when I actually cut this up, these like dotted lines here represent the spots at which that restriction enzyme cuts. So yes, it cuts out this gene really nicely, but I also get some other stretches of random DNA cut that I don't necessarily want, but I can't isolate this gene and like make it separate. It's not as nice and clean cut as this. So I can cut at various points, but since it's all mixed together now, the mixture of DNA that I have is a mixture of the gene I want and some other random parts of the DNA. So now I have them all together and I mix the plasmids with the frog, or in our case, the jellyfish DNA. And it inserts itself in there, boom, and we have this nice plasmid that has that gene that we want. But also, so again, what we want, but at the same time, some of the plasmids are going to take up those random sections of DNA because those restriction enzymes cut, yes, my gene, so I have some that are nice like this, but then I cut up, I had these other leftover sections of DNA, and I had more DNA, I had like the whole genome, so I'm going to get random sections. So some of my plasmids are going to have my gene I want, some are going to have random sections, and some are just going to close back in on themselves. So this is kind of hard to see here, but some are not going to take up anything and they're just going to get cut, open up, and then go back together. So we're going to have three different kind of types of plasmids that are produced. The one we want, the one that was transformed with the gene of interest. This one was technically transformed. It took on some genes or some DNA, but I don't really care about this DNA. And then I have some that just closed up on themselves. So cool. These are just genetic material though. This is just a plasmid. This isn't an actual organism. So what I do is I take some bacterial cells. I take some actual E. coli in the lab. So this is an E. coli cell. And notice it has its own little plasmid there. We're not going to mess with the plasmid that this bacteria has. We're just going to insert another plasmid so that it will transcribe and translate that DNA as well. So how it actually works is I take the bacterial cells and I heat shock them, essentially, is what it's called. So I heat them up really, really fast, and it forms holes within the cell membrane. So I'm getting some holes in the cell membrane of this bacterial cell, and so then when I mix these cells with the plasmids, those plasmids sneak in through the holes. So that's what she's showing here. And now I have a bacteria that has that transformed plasmid. And then the holes close back up because I make them cold, or I cool them down again, which closes those holes. So now I have some nice bacteria that have been transformed. This is the goal. But again, remember, I have some of these random ones that got the DNA that I didn't want. So this one is another transformed one, but I had that random DNA. And then some all have this bacterial cell that uptook this one that doesn't even have any extra DNA that I want. It's just another plasmid. Okay, so then what happens? How do you tell if it worked or not? So what we do is we take this bacteria and we plate it onto things called agar plates. So that's what she's doing right now. You can see, you might have seen these. They're like Petri dishes. Let me see if I can find one actually. I'm going to pause it. Okay, I'm back. So it's a little plate like this that has a lid. And the inside here you fill with agar, very similar to the gel electrophoresis stuff. So it has this gel on it. And the way that we make a few different kinds. So we make one that's just made out of nutrient broth, which is a place where bacteria grow. So we make it to where bacteria can grow and divide, replicate, do mitosis a bunch, so that they can make a lot more cells. And then some of them we mix in ampicillin, which is an antibiotic. So antibiotics kill bacteria. So why would we want to make some that could kill our bacteria? And I'll show you now how we figure out if our experiment was successful or not. So if I go back here, and you might have noticed this, but some of these, or all of them, have this antibiotic resistance gene. That's what this little red triangle is. So every single plasmid that I used is resistant to that ampicillin, meaning if I were to add it to the plates that had the ampicillin, they would be able to survive because they have this gene that is resistant to the antibiotic. So why is that important? Well, when I start to transform them, and I actually put them in to the cells, this plasmid has that antibiotic resistance gene. So this cell will survive and duplicate on my plate. So that's how I can tell, first of all, did it transform? Did it uptake a plasmid? And if it did, cool, it will survive. And that's how I know. I can know, A, it survived, it grew on here because it must have taken up that plasmid because the plasmid that it already had didn't have that antibiotic resistance gene. So if I put some bacteria onto this plate and let it like grow for a little while and I come back and nothing grew, that means it must have died. So that means that whatever I put on this plate didn't take up any plasmid because it only had its normal plasmid that's not resistant, so the antibiotic killed it. Then the ones that live, we know, okay, cool, they took up the plasmid because it has this antibiotic resistance. Every single one of these transformed plasmids has the antibiotic resistance. So that's how I can tell if I completed like some transformation or took it up at least. And then I can go and take the ones that grew and look at them under fluorescent UV light. And if I look at them under fluorescent UV light and they glow, that means that this is the one that it took up. If they survived but didn't glow, I either have this one here that has the random DNA or that one that doesn't have any of the cool things and is just the regular plasmid with no DNA, but I still have this antibiotic resistance. So some data would look like this. These ones both got the transformed bacteria. These ones both lived. So they both had colonies that look like these little dots on them. So they both took up some plasmid because they both got the antibiotic resistant gene and they both survived. But when I look at them under UV light, only this one glowed. So this one must have gotten a plasmid that took up the specific gene that I wanted. This one either didn't take up any of it and just closed back up on itself, but still got the antibiotic resistance, or it took up some of those random pieces of DNA that still got cut up. Cool?